So last time we finished with the discussion of red blood cells, right? We just few reminders, okay? We went over the composition of a blood. You okay? Composition of a blood, formed elements, which are cells, basically red blood cells, white blood cells, and platelets, and plasma, right? And then we talked about red blood cells. How their function is to transport oxygen in the blood. Oxygen binds to molecules of heme in the molecule of hemoglobin. So 4 molecules of heme. So there are 4 molecules of heme on the hemoglobin molecule, and each of these heme molecules can carry 1 oxygen. Okay, and hemoglobin becomes oxyhemoglobin in the lungs and unloads and delivers oxygen to the tissues. Now, when we had a conversation a little bit about erythropoiesis, let's talk about it a little bit more. So, the main stimulus for erythropoiesis. Poiesis means production, erythro means red blood cells, is hypoxia, low oxygen levels. Hypoxia can be caused by, for instance, lower levels of oxygen in the air. If you go to the mountains, you will definitely experience some hypoxia, and that will trigger the production of the hormone called erythropoietin. Okay, erythropoietin produced in kidneys. Via blood, erythropoietin is delivered to the bone marrow, and in the bone marrow, erythropoietin stimulates production of new red blood cells. New red blood cells start to deliver more oxygen to the tissues, including kidneys. No hypoxia anymore. You're fine. This is the long-term adjustment to hypoxia, you have to understand. So it doesn't take 10 minutes. It takes a few days, actually. And to get completely adjusted, it takes a while. Okay? Other possible sources. Well, hypoxia may be sort of healthy because you are in the mountains. Or it can be unhealthy if your respiratory function is impaired. Ability of your blood to deliver oxygen to the tissues may be affected by the number of available functional red blood cells, which means anemia. Iron deficient anemia will lead to production of more red blood cells. Does that make sense? All kinds of anemias will lead to increased red blood cell production. But you have to understand that if we do not have enough iron, then new red blood cells won't be functional. And if we have aplastic anemia or pernicious anemia, new red blood cells won't be functional. They will not mature properly. Iron is a necessary component for proper red blood cell function. It is stored in complex with proteins and transported from liver. It's stored mostly in the liver, transported from liver to the bone marrow in the complex with the protein called transferrin. We do lose we do lose iron a little every day, so it has to be replenished with the diet. It is more critical in females of the menstrual age because there's a monthly blood loss, and in this case iron has to be replenished. It's more critical. Excessive iron consumption, like if you decide to start taking iron supplements, will not lead to the higher red blood cell levels. Does that make sense? So hoping to improve your aerobic endurance by consuming copious amounts of iron is the false hope. Vitamins that are necessary for proper erythropoiesis are vitamins B12 and folic acid. We usually we receive both those vitamins in sufficient amounts from diet. Folic acid now is added to a massive number of foods, just to provide this vitamin to kids, because this vitamin is important for growing, replicating cells. Okay, so kids, pregnant women, they have higher demand for this vitamin. Vitamin B12, we need this vitamin in such small amounts that we easily get it from the diet. The main problem with B12 is that we do not absorb it. Not we, but some patients that do not properly absorb vitamin B12, they develop a condition known as pernicious anemia, and they do not have enough red blood cells because they do not divide or they do not differentiate in the proper fashion. What is the reason why iron is not present in the blood in its free form? One of the main evolutionary reasons, as some scientists think, is that bacteria also need iron. Many of them need iron. And if iron would be available in a free form, any simple bacterial infection that would make its way to the blood would immediately kill us, just because bacteria would replicate to such an enormous amount. These proteins, siderin, transferrin, they hold on to iron in a way that bacteria cannot pry it off. Do you understand what I'm saying? Iron is attached to those proteins very tightly. Let's put it this way. Okay. Red blood cells have a limit in terms of the lifespan, about 3 to 4 months. Once red blood cells are old, they are brought to the spleen, mostly to the spleen, also to the liver and bone marrow, where red blood cells are destroyed. Iron is recycled, and heme is, as we discussed before, ends up in the bile. From the bile it enters it is converted into bilirubin. Bilirubin ends up in the bile. Bilirubin enters the small intestine, and then it's either excreted with the feces or converted into the urobilinogen, and then excreted in the feces and urine. That makes sense. What I really want you to focus on, seriously speaking, is to know pretty well how erythropoiesis works. Hypoxia, erythropoiesis. Anemia, erythropoiesis. Iron deficiency, erythropoiesis. And of course, I could not pass on the opportunity to say about erythropoietin as the performance enhancement drug. Nobody uses it anymore, really, because it's very easily detected in the blood, but up to some point it was great. What would you and imagine you have an athlete, say a runner, starts to use erythropoietin as the performance enhancement drug. So when a person injects erythropoietin, what happens to the number of red blood cells? It increases, yeah. What happens to the viscosity of the blood? It increases. What happens to the blood pressure? It increases, which increases the risk of stroke. And just simply the presence of red blood cells increases the risk for clots. It was pretty common condition, pretty common complication in especially cyclists, because they were abusing this massively. Another great way to kind of circumvent this is to get blood transfusions. Just simply get extra erythrocytes, extra red blood cells. Yes?
So is that
Blood doping is blood transfusions. Yep. Yep. That's totally illegal. And there was something, they did something to circumvent the checkpoints. I know that Armstrong had, you know, what's Neil Armstrong, what's his name? Lance Armstrong, whatever. Neil Armstrong was a different dude. There's so many Armstrongs here. Anyway, Lance, he had sort of an illegal supply of blood donors. So they, on the way to the race, they stopped in the forest, did a transfusion, and just moved on. So he had like a lab in the bus, proper. It was all sanitary. It was all very clean and nice. It was totally illegal. So legal doping is living in Colorado. That's it. And then competing not in Colorado. That's important. Because if you live in Colorado and then you go to Europe and compete in Alps, there's no change. There's the same condition. But if you live in Colorado and then go somewhere, you know, flat, like, I don't know, Texas, different parts of Texas, any flat land, and you compete there, then you definitely have an advantage. Okay. Now, disorders associated with red blood cells. The most famous is the sickle cell anemia. Single mutation, one mutation in one nucleotide in the gene for hemoglobin turns normal hemoglobin A into hemoglobin S. When concentration of oxygen is low, which means in the tissues it's lower, right? When concentration of oxygen is lower, hemoglobin S starts to polymerize. And when it polymerizes, changes the shape of the red blood cells to sickle shape. These cells are fragile, first of all, so they get broken down, which leads to hemolysis. They also damage endothelium because just by looking at those two shapes, if you think about it, this cell, you can appreciate, sorry, it can fairly easily pass through the blood vessel. It's squeezable. It's like a ball, really, you know. If this thing with sharp corners will be squeezing through the blood vessel, it will damage the endothelium. And it may lead to the increased permeability, inflammation, vasculitis, increased risk for clots, and so on and so forth. Does that make sense? So basically, treatments. Okay, which treatments are available? First, blood transfusions. Okay, for many patients with sickle cell anemia, blood transfusion is the only long-term hope. The main problem with blood transfusion is that after every blood transfusion, the recipient experiences hemolysis, breakdown of red blood cells. That is normal. When you donate, you don't experience. Don't take me wrong. There are going to be like donation here at Lakeland Wednesday, Thursday. Do. Okay. So, what I was talking about? Oh yeah, hemolysis. Red blood cells disintegrate, and you have iron going into the blood, which ends up in the kidneys. Iron is toxic for kidneys. Kidneys eventually shut down, and that is often a case, the kidney failure, pretty much a case of mortality or morbidity for patients with sickle cell anemia. Now, this disease can also be treated by hydroxyurea, which stimulates the production of hemoglobin F, fetal hemoglobin, which does not turn into hemoglobin S at the low oxygen concentration conditions. Okay? So this condition is treatable. It's not curable. Technically, you can do a bone marrow transplant, but it's warranted only in the severe cases because you render a person, it's hard. It's hard. We're going to talk about transplantation. There are a lot of complications associated with it. So that's one. Thalassemia is a condition in which people have just lower levels of red blood cells and lower levels of hemoglobin in those red blood cells. So their tissues experience hypoxia. Linked traits are the various bone deformities, splenomegaly, the cardiac abnormalities. Treatments are the same: blood transfusions and bone marrow transplant. But what's interesting, if you would look at the areas where these conditions are common, sickle cell anemia stems from Central Africa. Okay? Thalassemia, I don't have this map here, but trust me. Thalassemia is most common in the Mediterranean region, countries that surround Mediterranean Sea. Both those areas, Mediterranean region and sub-Saharan Africa, this part of Africa, known for the presence of malaria. Turns out that both these conditions protect the carriers from malaria. And for sickle cell anemia, what happens is, in people with functional red blood cells, malaria parasite can successfully complete its life cycle, producing more parasites, causing the actual disease, malarial disease, and spreading from one person to another. In people with sickle-shaped red blood cells, malarial parasite cannot complete the life cycle. And since sickle-shaped red blood cell is not normal, then macrophages will eventually destroy them with the parasite. Does that make sense? And I think it's pretty awesome. Moreover, since parasite infects red blood cells, it decreases the amount of oxygen in each red blood cell. So infected red blood cells become sickle-shaped. And they basically selectively destroyed. So parasite infects cells, changes their shape immediately, macrophages immediately take care of them. Now, if we wouldn't have this piece of information, we would probably say it would be, that's a great example of how the trait that is not beneficial, and sickle cell anemia is the genetic disorder, how this trait is maintained in the population. Let me explain. If there is a kid with sickle cell anemia, sickle cell anemia is the recessive disorder, which means the kid has to have two recessive genes that would encode for the mutant hemoglobin. Does that make sense? Do you understand the idea of recessive and dominant? So two recessive. Is this kid going to live a good life? No, it's going to be terrible. Okay? And if we would not provide treatments to those kids, they would die before they reach reproductive age. Yes?
Are you saying which one comes first? Malaria, the sickle cell, and the other one, are they the natural disformities against?
Normal red blood cells get infected with malaria and they become deformed. And since they become deformed, the immune system destroys them. Does that make sense? If they're not infected, they do not become sickle cell shaped. Better? Does that make sense? Only if they're infected, they become sickle cell shaped in people with that gene. So people who have actual disease, they have two recessive alleles, they horribly sick, and especially in Africa, chances of those kids to make it to the reproductive age are low. Now, kids who have one dominant and one recessive gene, so normal hemoglobin and bad hemoglobin, they're not sick with sickle cell anemia. They do not have that sickle cell shape of red blood cells. So they're healthy, they're normal. Does that make sense? When they become infected with malaria, their chances for survival are much higher than in people who do not have the gene at all. People who, like AA, people who have no genetic, no hereditary info about sickle cell anemia. Does that make sense? So, presence of this gene, turns out, doesn't confer any disadvantage, but provides an enormous advantage which can be seen only in this part of Africa. Does that make sense? Here's another interesting story. If you think about it, here in the United States, having a gene for sickle cell anemia provides absolutely no evolutionary advantage. Why? There's no malaria in the US. Does that make sense? So only in Africa, and it will remain there as long as malaria is present there. Okay? Same goes for, well, Mediterranean, slightly different story. Mosquito control efforts reduced the number of mosquitoes a lot, so malaria transmission in the Mediterranean region went down tremendously. Okay? Thalassemia remains more of a genetic disorder, and I don't want to sound like a Nazi, and no, seriously, and I understand that you may know people with those conditions, but it's sort of paradoxical that when these people receive treatment, we improve their quality of life and we improve their longevity, but at the same time we weaken the genetic pool. And we have just to be conscious about it. Okay? You have to understand that a lot of genes that otherwise would be eliminated from the population, we allow them to persist. Does that make sense? Now, white blood cells are cells that are responsible for various types of immune responses, immune protection. And there's not a lot of them, okay? But this is what, so let me put it this way. When you go to the doctor and they do cell count, they count red blood cells, they count platelets, they count white blood cells. Now, red blood cells number tells the doctor if your bone marrow produces enough erythrocytes to carry oxygen around. Platelet count tells your doctor if the thrombus formation process works fine. Okay? Also, number of platelets may tell doctor about any hidden infectious processes that occur in your blood vessels. There's clotting somewhere. And white blood cells. Now, imbalances in the number of white blood cells may tell the physician that some sort of infectious process is going on. All white blood cells can be divided into the granulocytes and agranulocytes. You can see that all granulocytic cells have granules in the cytoplasm. Agranulocytes, as you can guess from the name, have no granules. Okay? Now, functionally, let's talk about agranulocytes first. Lymphocytes. The word lympho suggests that you're going to find these cells where? In which system? In the lymphatic system. Well, anything that's in the lymphatic system can make its way to the blood, right? So you're actually going to find them in both, but those are the main immune cells found in the lymphatic system. They're rather small. The appearance is very distinctive. They're small with a huge nucleus which fills up practically the entire cell. Okay? And lymphocytes are the main responders against virus infections or bacterial infections. They largely responsible for leukocytosis. However, I must say, if you do differential cell count and you find a lot of lymphocytes, it is suggestive of virus infection, mostly. Monocytes, very interesting cells. They're large, they're biggest white blood cells with crescent-shaped nucleus, and by themselves, monocytes are not functional. They can be found in a circulation, okay, in the blood. They respond to the inflammation in the tissues. If there is some kind of an infection, a damage, an injury to the tissue, monocytes will be able to sense this and migrate into the tissue and differentiate, become a macrophage. Does that make sense? I want you to understand it very clearly. So monocytes, they circulate. They're like Vikings, okay? They walk around, and when they see injury, they turn on berserk mode and rush into the tissue and kill everything. Monocyte by itself isn't really doing a lot. But when they proliferate, when they differentiate, they become macrophages then. They become eaters. Does that make sense? And they're really not specific. They eat everything. From bacteria to parasitic infections to viruses, they would eat everything. Debris. If you would have a bruise, some sort of a deep bruise, which is technically sterile, there's going to be a lot of macrophages around that bruise, eating the dead cells. Does that make sense? Now, those were two types of agranulocytes. Granulocytes can be generally broken down to three categories. First one is neutrophils. Neutrophils got their name because they can be stained by both acidic and basic dyes. So they're kind of neutral. Okay? They have multi-lobe nucleus, and those are the largest, not the largest, the most numerous white blood cells. The main target for neutrophils are bacterial cells. What they can do? They can phagocytose, first of all. But neutrophils are disposable heroes, basically. When macrophages can eat and eat and eat and phagocytose and keep going, neutrophils are, as one of my students said once, one and done. They phagocytose and die. Other methods of warfare that they can employ, look, this is just, you think all of it is old stuff? This, what I'm going to tell you, this property of neutrophils, was discovered in 2004. So it's not that old. It's called NET. NET is neutrophil extracellular trap. What they do when neutrophils die, they may release the DNA, which is a pretty long molecule, right? DNA will wrap around bacteria and will hold it in place. And then granules, neutrophil granules, those little tiny bubbles, they contain different oxidative chemicals. Basically, if you think about it, they contain bleach. Okay? It's called oxidative burst. And those chemicals will be spilled on the bacteria that are already trapped, and bacteria will die. Does that make sense? I think it's incredibly elegant. Okay? When you have some sort of an infection, say skin infection, it's a good example. Pus. This whitish. This white stuff, dead neutrophils. The majority of it is really dead neutrophils. Okay. So if you have a patient who shows it's not neutropenia. Well, we don't like elevated number of neutrophils. And this patient most likely has bacterial infection. Okay, does that make sense? Eosinophils. Eosinophils can be stained with acidic dyes, hence the name. Their primary function is the response to parasitic infections, from single-celled parasites to worms. In fact, one of the, when there's a patient that has complained some kind of infection, and doctors do the blood count and they find that eosinophils are elevated, that's the first sign we need to check for worms of any sorts. Does that make sense? You can imagine that if you have a worm, which worm can you get in the United States? If you live here and don't travel much, then unless you live in rural Alabama, then your worst nightmare can be probably Toxocara canis. It's the dog heartworm, which can acquire, it can be pretty deadly for humans because it's rare. But that's it. It will cause elevated eosinophil counts. You can treat it, but the point that I'm trying to make is that even Toxocara are big animals. By big, I mean half an inch long. Eosinophil cannot phagocytose the worm, right? So what they do instead, they release toxins and chemicals of oxidative burst to either poison the worm or to bug the hell out of it. It works especially well with the worms that can be found in the intestine. Think about this. If you have an intestinal lumen, right, and the worm is sitting inside of the intestine, and it's attached to the intestinal surface, it causes local inflammation. Eosinophils migrate there and start to produce chemicals. And it bothers the worm. So what can happen, it'll detach and it will be pussed with feces. Okay? And there are examples of it. Yes?
The chemicals that they release hurt, the person, or does it just hurt the bacteria?
It's a great question. Well, they can, yes. Generally, inflammatory response can damage the tissue, of course, yeah. But we have a lot of cells. If we kill a few thousand, nobody cares. It's okay. As long as this inflammation eventually subsides. Does that make sense? So that's eosinophils. They bug the heck out of the worm, and the worm leaves or migrates somehow. Basophils are main regulators of inflammation. They produce histamine mostly. Okay? When you get some sort of an injury, let's say bruise, okay? How does it look? How does the area look? Black and blue, usually swollen, right? Sometimes, you can see like around that black and blue where the blood vessels burst, you can see red because of the inflammation. That red increased blood flow to the site of injury is because of histamine that basophils release. The word histamine, does it sound familiar to you? Well, not only from digestive system, just for day-to-day life. When you take antihistamines, what do you treat? Allergies. Basophils can become so-called mast cells. Mast cells are basically the same basophils, but these guys are sitting in the tissue. Does that make sense? And it's normal, you have a lot of mast cells in your mucous membranes. So now let's say I'm allergic. I really allergic to cats. Let's say I pet a cat, okay? And then I touch my nose. Which means that whatever allergen is in the cat's skin, this allergen gets into my nose and interacts with the receptors on the mast cells that are in the lamina propria of the mucous membrane of the nose. And mast cells, which are basically those same basophils, they start to degranulate. They start to release the content of the granules. That makes sense. Granules contain copious amounts of histamine. Histamine causes massive vasodilation in my mucous membrane, which leads to a lot of nasal secretion, and I have sniffles. I have runny nose, allergic rhinitis. Does that make sense? If somehow all mast cells in the entire human body are stimulated by an allergen, all of them release histamine, and all blood vessels immediately dilate. What's going to happen to the blood pressure? It's going to drop precipitously, and we call it vascular shock. That makes sense? Part of anaphylactic shock. So, leukopoiesis. First and foremost, I think I mentioned it before. I am not asking you to memorize all the steps. For instance, all I want you to understand, all blood cells, no exceptions, originate in the bone marrow. Okay? From the same hematopoietic stem cell. Hematopoietic stem cell will become red blood cell, as we discussed, and it will become white blood cell. But interesting thing is that hematopoietic stem cell can differentiate. We're all familiar and comfortable with the word differentiate, right? What does that mean, differentiate? Not just change. Change how? Become more specialized. Become more specialized. So hematopoietic stem cell can differentiate into two lineages, myeloid and lymphoid. Myeloid stem cell, myeloid lineage, it gives rise to all three granulocytes and monocytes. Does that make sense? So monocytes and all three granulocytic cells are cells of myeloid lineage. Myelo means blood. Makes perfect sense because they are normally found in blood. We good? Lymphoid lineage is represented by lymphocytes. So far, let me kind of summarize it for you. We've got red blood cells that are produced from hematopoietic stem cells when there is a hypoxia, right? Of any cause. White blood cells produced from the same hematopoietic stem cells, but they can form two lineages. Myeloid lineage, represented by granulocytes and monocyte, and lymphoid lineage, represented by lymphocytes. You don't need to memorize all those terrible steps. Okay? What would trigger formation of those cells? What is the function of white blood cells? What do they do? Protect. So if there is something to protect, that's the stimulus. Does that make sense? So basically, leukocytosis is the signal that there's some kind of an infection going on. Good? Clear? And when infection is gone, the number of white blood cells essentially decreases. Progression of this leukopoiesis is controlled, sort of, by the various colony-stimulating factors, interleukins, various molecules. It's actually chemically controlled. If you think, a cell can be infected, it will produce interleukins. Interleukins will, say, stimulate neutrophils to be the stem cells to become neutrophils. So you've got a lot of neutrophils. You have an infection with the worm, different interleukins are produced, so now you get a lot of eosinophils instead of neutrophils. That make sense? So, one of my favorite parts to talk actually about an infection and inflammation process. Is inflammation normal? I got one affirmative answer. Is it normal? Inflammation? Like if I just tell you, inflammation, is it normal? What do you think? Yeah, it's normal like in general. If you don't develop inflammation, you're screwed. Seriously. Something is wrong with you. Okay? Uncontrollable inflammation is bad. That's a different story. So what happens when a person, has a tissue injury, whether it's the infection or any kind of other cellular damage? Let's talk about infection, okay? So infection. Cells that are affected by this injury, they start to produce cytokines, like interleukins. And interleukins from the cells enter the bloodstream. And they end up in the bone marrow. Does that make sense? And in the bone marrow, they will stimulate leukocytosis. It's first and foremost. So leukocytosis means what? A lot of leukocytosis. A lot of white leukocytes. A lot of leukocytes. Yeah, white blood cells. Leukocytes. Perfect. So now these leukocytes, they enter the bloodstream. They're all pumped up. They're ready to kill whatever they need to kill. There's a little problem. The problem is, where are they supposed to go? So they travel through the blood vessels and they search the molecules called adhesion molecules. Okay? Does that make sense? So far, do you follow me? What do you think adhesion molecules do? All in the name. Stick. They allow white blood cells to stick. Clear? What essentially happens, say, there is some kind of an injury right here. Injured cells will produce interleukins. Interleukins will increase number of adhesion molecules on the endothelium. White blood cells, believe me, randomly will bump into those adhesion molecules and will stick to them. Can they detach? Sure. If they don't, they will start to roll. So first is rolling. Then some of them will firmly adhere, and eventually they will start to migrate across the vessel wall. The migration process is slow. I never managed to see the entire. I didn't have enough patience. When I watched the mouse, it may take like hours. Never had enough patience just for kicks to watch the migration. But basically what happens, white blood cell, can squeeze its way between the endothelial cells. This is called paracellular diapedesis. Perfect name for the arthro band. Okay? Paracellular means between the cells. Diapedesis in general means movement through the wall. Okay? In some cases, cells can travel through a cell, through the pore in endothelial cell. This is called transcellular diapedesis. Okay, so diapedesis is the migration of white blood cell across the wall of the vessel. Does that make sense? Now, white blood cell can go into the tissue, but sometimes it returns back into the blood. So a couple more terms. When it goes out into the tissue, this is extravasation. When it returns back into the blood, this is intravasation. Obviously, we are more interested in getting into the tissue. Right. So, a little problem is that generally endothelium, especially if you think about it, endothelium in continuous capillaries or in the postcapillary venules, endothelial cells are connected to each other by tight junctions. It's hard to squeeze between. Turns out, interleukins, those inflammatory mediators, cytokines, they will also increase vascular permeability, allowing white blood cells to squeeze between the endothelial cells. Okay? So now we have white blood cells in the tissue. They're not at the injury site yet. How do they find it? They follow chemical cues, so-called positive chemotaxis. As you can imagine, the highest concentration of cytokines and other inflammatory molecules will be at the site of injury. And then it will gradually reduce. Does that make sense here? So white blood cells will slowly migrate towards the injured site. Okay? And on their way, monocytes will become macrophages. Mostly two cells, monocytes and macrophages and neutrophils will make their way to the injury site. They will kill whatever is supposed to be killed, take care of the debris, kill microbes, do all the regular job, right? Another thing that will spill into the tissue is plasma. What do you think is the function of the plasma flow, fluid flow, into the site of injury? It's a massive flow of fluid to the place where there's a lot of crap to wash it off. Exactly. All this crap, cellular crap, bacteria and everything else, will be moved into the lymphatic system to either completely recycle it or build the immune response, stuff like that. Okay? Another important feature. Mentioned before that plasma contains a protein called fibrin. Well, technically it's fibrinogen. Okay, fibrinogen becomes fibrin eventually. But the point is, when plasma enters the tissue, fibrinogen can get activated and become fibrin. Fibrin will form long threads, and it will make sort of a net that will prevent potential microbes, microbes, from potentially spreading away. Does that make sense here? Cool? Now, it's all pretty awesome, right? But, okay, let's think about this. If you have an injury, sorry, I ate something wrong yesterday, and I'm stuffed with anti-diarrheal. Sorry for the detail, but I'm like on the pills up to this level. And the only thing that I ate since yesterday is the bag of sort of like bread, you know, this Snyder's thing. So I'm kind of want to see it sometimes. Now think about this. You have a bump. I believe the thoughts that you have when you look at the bump, or I don't know, you have a sprain or something, are not exactly about migration of white blood cells and increased vascular permeability and clotting factors that, you know, keep all the debris in place. What do you think? What do you have? What do you see? What are the signs of the injury? Swelling. Why? Why does it swell? You know everything, I told you. Why does it swell? Why does it become larger? Exactly. Plasma leakage. Good. Swelling. What else? Redness. Why? Rush of blood and dilation of blood vessels. Exactly. What else? Huh? Heat. Well, yeah, it's basically it's chemical. Those cytokines, they will increase the temperature. What's the fourth sign? Yes. Why? Two reasons. Excess pressure from the swelling and chemicals released from the damaged tissue. So in Latin it's going to be rhubarb, color, tumor, and dolor. If you want to, you know, I can write it down for you. So rhubarb means red, okay, like ruby. Color means temperature. Tumor means swelling. And dolor means pain. Does that make sense? And we just explained why, how these four signs of inflammation develop. Does that make sense? So I want you to be comfortable with the steps of inflammation. In the study guide, I basically guide you through the question-answer manner what you need to know. Platelets. I like to mess with it and say from time to time that platelets are merely the bags of cytoplasm. I have a good friend, she works on physiology of platelets. She would kill me over this. For a long time, platelets were thought to be really bags of cytoplasm. How are they formed? The hematopoietic stem cell develops into the cell called megakaryocyte. And then megakaryocyte produces this protrusions. Basically, platelets are just pinching off the surface of megakaryocyte. You see that? So, yeah, they are bags of cytoplasm. What is interesting about platelets is that they contain RNA, messenger RNA, and they have active gene expression. So they're not that simple after all. They do not have nucleus, don't get me wrong. They do not have mitochondria. But they do have ribosomes, and they do have RNA, and they do produce proteins. They do have expression. So, for a long time, platelets were thought to form clot. That's it. Period. It turns out, though, that they're much more than that. Platelets are critical regulators of inflammatory responses. Turns out that platelets in the blood vessel with inflammation and blood vessel without inflammation, they're going to look different. Platelets can be activated by the inflammation, which opens up a whole field of opportunities if you think about it. People develop strokes, which is blood clot that ends up in the cerebral circulation. This blood clot usually comes from where? Most frequently. Do you know where those clots are formed? Most frequently. In people with healthy hearts. Some clots are formed in the during the atrial fibrillation. That's one opportunity. But what is the most common DVT? Deep vein thrombosis. Peripheral veins, mostly legs. Two major reasons. One, the blood flow is kind of stagnant, especially if you sit for a long time. Second, usually in people who are obese and have diabetes, the peripheral circulation is impaired. There's endothelial damage or some other kind of a vascular damage. Vascular damage means vascular inflammation. Vascular inflammation means platelet activation. Platelet activation means clot. Do I make sense? So you have to understand clotting is good. No, thrombus formation is good, but when it's not properly regulated, then may lead to some bad consequences. So platelet formation is regulated by the hormone that is called thrombopoietin, and it's really fascinating. So thrombopoietin, molecule of thrombopoietin, binds to platelets. So this is the bound thrombopoietin. And if you have a lot of platelets, then you have very little free thrombopoietin. That makes sense? When platelets start to form clots, or there's any kind of condition that reduces the number of platelets, you found yourself with a lot of free thrombopoietin. Okay? And that immediately initiates the platelet production. Thrombopoietin becomes free, acts on the bone marrow, stimulates platelet production. You have more platelets. Platelets bind thrombopoietin. You have no free hormone. Does that make sense? It's pretty elegant. So platelets are essentially the key players in the process called hemostasis. Hemo, blood, stasis, balance. Hemostasis specifically refers to the process that prevents blood loss. Blood loss due to the injury to the blood vessel. Does that make sense? So what happens? What are the steps? So imagine there is some kind of a blood vessel injury. Okay? Injury to the blood vessel, which it will immediately produce huge number of chemicals, of which you know some. Epinephrine. You're You've heard serotonin. You didn't hear, I believe, thromboxane A. It's another really important chemical, which we're going to mention pretty soon. So all those chemicals, and they all initiate vasoconstriction, vascular spasm. Makes perfect sense if you think about it. If you have a leaky pipe, you want to turn off the water first and foremost. Okay? In the body, we can do it locally. We can do vasoconstriction. Make sense? So that's first step of hemostasis. Vascular spasm. Damage. Damage to the vessel wall. It means that endothelial cells are gone and underlying collagen, which comprises a huge portion of the basement membrane, is exposed in the blood. Platelets have receptors that can interact with the collagen. Integrin receptors, most famous is von Willebrand factor. If you will be working in oncology wards or with patients who have various blood abnormalities, you may encounter patients with von Willebrand deficiency. In these patients, when platelets are exposed to collagen, these platelets do not have von Willebrand factor. So they do not attach to collagen, and they do not form platelet plug. Does that make sense? So they're supposed to stick to the damaged area, start piling one on another, but they don't. And those people suffer from excessive bleeding disorders. That makes sense? Platelets simply sticking to collagen is not enough. You need to pile them on. So platelets get activated by the same thromboxane A and adenosine diphosphate, or ADP, from endothelial cells. All these chemicals start to activate platelets. Platelets get activated, stick to the plug, produce chemicals, activate more platelets, which stick to the plug, produce chemicals, activate more platelets, and it becomes basically a system with a positive feedback. In the meantime, I want to find a video. I just recollected that I have one. Oh yeah, I'm going to show it to you later. I have it embedded. Perfect. So it's a system with positive feedback. Does that make sense? Platelets get activated, stick to the plug, release more chemicals which activate more platelets which stick to the plug. So, and eventually you have a platelet plug formed. Does that make sense? Now, you think for a second about positive feedback. If just, with that information I just gave you, what should happen? You should die in like 2 minutes because of all platelets being activated and just making a giant clot everywhere in the body. One of the reasons it doesn't happen, areas of the blood vessel, like this one, that are not affected by injury, they produce nitric oxide and prostacyclin, chemicals that actually prevent platelet activation. Does that make sense? So platelets get activated only at the site of injury. Anywhere else, they aren't. That makes sense? Now think about this. You have a brick wall. and then some media drove through it. Now I have a gap in the brick wall. You need to fix it. So bring in the truck of bricks. Start putting bricks, one by one, and you fill it up. You do exactly what I just told. Just you put bricks, okay? Brick, another brick, another brick. Is that going to be a good repair? What are you missing? Mortar. You're missing mortar. So in any good platelet plug, you need something that would hold platelets together. You need those fibrin threads from plasma. In addition to platelet plug, you need coagulation. Coagulation can be stimulated by two pathways: intrinsic and extrinsic. Words, what do words intrinsic, extrinsic, what do they mean? Something is intrinsic. Well, it even sounds like inside. Yeah, and extrinsic is outside. So intrinsic means that the stimulus is sort of contained in the blood vessel. Collagen. Most commonly collagen. So think about this, if there's a damage to the endothelial cells, let me go back for a second. There's a damage to endothelial cells, then this collagen here will be exposed to the blood. Do I make sense? Can you see that? So this layer of collagen, if endothelial cells are gone, it will be exposed to the blood. And in addition to platelets, it will activate the intrinsic pathway. Now, both intrinsic and extrinsic pathway, they are cascade pathways, like a domino effect. One coagulation factor becomes activated, and it converts next one into its active form. So the next one converts the third one, and the third one converts the fourth and fifth, and so on and so forth. Does that make sense? Numbering doesn't make much sense at all because they were numbered in the order they were discovered. So in intrinsic pathway, it's better than you think. It's not as bad in terms of, step memorization. So what I want you to understand, just steps. So 12 activates 11. Now here's the gap. 11 activates 9. 9 activates 8. And they form complex. Okay? So far we're missing 10. We're good? Now, what is the difference between intrinsic and extrinsic? In extrinsic pathway, activation comes out from the tissue. Tissue contains so-called tissue factor, which activates 7. They form a complex, and now look at this. This is super elegant. On the intrinsic side, you have a complex of 8 and 9. On the extrinsic, you have a complex of 7 and tissue factor. Either of those will convert factor number 10 into its active form. Does that make sense so far? So think about this. 12, 11, 9, 8. 7, and that's it. 7 and tissue factor, that's it. They converge on the same point and activate factor 10. Factor 10 activates number 5. Don't memorize number 5. What I want you to understand, they converge on number 10. Okay? And that number 10, along with few other molecules, calcium, phosphatidylserine, and factor 5, they form a molecule called prothrombin activator. Before we move on, I want to ask you a few questions. You have no idea, and you will be able to answer this question right now. If you look at the name, what's the function of prothrombin activator? What does it do? It's all in the name. Prothrombin activator. It activates thrombin. That activates prothrombin. Yeah, that's it. That's all you need to know. Period. It's good. Okay. That makes sense? Another question that I want to ask you. Which pathway, intrinsic or extrinsic, is faster? Extrinsic. Why? Fewer steps. Any evolutionary reasoning behind it? Why should it be faster? Huh? What about injury? So, what is this? Where does the stimulus for extrinsic pathway comes from? But no, extrinsic. Where does the stimulus come from? Is it vessel or something more than vessel? Is it in the vessel or beyond the vessel? Extrinsic pathway. Beyond the vessel. It's tissue. So basically, if we have an injury as far as tissue, we should hurry. If it's blood vessel, we don't have to. We can kind of take it slow. Does that make sense? Okay. So we've got 12, 11, 9, and 8 on the intrinsic, 7 in tissue factor on extrinsic. They all converge. Factor 10 becomes a crucial molecule in the prothrombin activator. Prothrombin activator, guess what? Activates prothrombin and makes thrombin. Thrombin becomes thrombin. Thrombin activates fibrinogen. Fibrinogen becomes fibrin. Fibrin forms threads. Threads form mesh. Mesh holds red blood cells together. Does that make sense? I think it's kind of straightforward, isn't it? So, steps. I specifically tell you, don't focus on factor 5 because it kind of throws the entire scheme of things away. I just know that. Know that calcium is needed. Calcium is really, really needed. Without calcium, there's no coagulation. Does that make sense? And know what triggers intrinsic and extrinsic factors. Extrinsic damages outside of the blood vessel. Intrinsic, the damages inside of the blood vessel. Now, let me show you. Well, actually, you know what? Let's talk about, first of all, how clot is controlled. So, okay, we have a damage. That's the damage, okay? We have a clot. We have fibrin holding it together. Eventually, platelets start to squeeze the plasma out of the clot, and they will bring the ends of the injury closer. Platelets will produce growth factors, so-called platelet-derived growth factors, that will restore the vessel wall. Smooth muscles and connective tissue. Endothelial growth factor, VEGF. will stimulate growth of endothelial cells. Okay? So eventually, the walls of the vessel will be restored. You still have a clot, and you need to get rid of it. This is achieved by the release of tissue plasminogen activator. Tissue plasminogen activator activates plasminogen, which becomes plasmin, and plasmin degrades fibrin. Does that make sense? So healthy endothelium that is restored at the site of the clot starts to produce tissue plasminogen activator. It destroys the fibrin mesh. Fibrin mesh is gone. Platelets are gone. Are we clear? Why clot doesn't excessively grow? Clot is positive feedback, right? A few things. First, blood flow. Blood simply takes away excessive growth factors. It dilutes it. You know, the solution to pollution is dilution. So accumulation of those molecules that can activate platelets, blood just takes them away. Okay. As I mentioned, nitric oxide and prostacyclin prevent platelet activation over the healthy endothelium. Okay. Thrombin binds to fibrin. So the more fibrin is produced, the less thrombin you're going to have in the blood. Do I make sense? As fibrin accumulates, it binds thrombin away. It kind of reduces clot growth by itself. Okay? There are molecules that directly control, that directly inhibit thrombin, like antithrombin or heparin. They directly inhibit thrombin activity, kind of keeping it in check. Now this video, okay, just 1 second, let me try it. Okay, awesome, it works. Can you see that now? Let me turn off the light. So this video was taken by a colleague of mine, and it shows the formation of the blood clot in the arteriole, the blood vessel, and as far as I can see, it's about 30 micrometers in diameter. And the cremaster muscle, the muscle that holds testicles, of a mouse. Okay? In this case, clotting process is induced by exposing the blood vessel to the light. Mouse, before this, mouse receives a chemical that is light activated. When chemical that's circulating in the blood is exposed to the light, this chemical causes damage to endothelium, essentially triggering platelet activation and clotting via intrinsic pathway. Does that make sense? It takes a lot of time. And this is sort of a speed up example. So you can kind of see the the blood flow. I will have to use this. So it's kind of a lot of skipping going on. And look at the formation, accumulation of white clot. So you see it constricts. That's the first response to the damage. That's vasospasm. Okay? This. Can you see this little accumulation? Used to be here, but it kind of went away. Here. Those are platelets. Those are platelets. Those are platelets. Those are all platelets accumulating But you see how blood removes them? Because it's really fast. And it just can't stick. There's not enough activation. So it's not sustainable yet. A lot of skipping going on. And finally, you see the massive accumulation. And then, at some point, it's like an avalanche. It's stopped. That's it. The blood flow ceases immediately. Okay? Now, this will never go back. The particular arteriole is done. Okay? I just want, so first of all, please, I beg you, don't memorize the factors with, is it a protein, is it an ion, you know, what's the number 7, number 8, what's the name of it. One basically take-home message, there's a lot, right? But one take-home message is that deficiency in the factors results in a condition known as hemophilia. And this condition became famous via royal families in Europe, which were all very closely related. For instance, last Russian emperor and German emperor during the First World War, they were cousins. They wrote letters to each other, talking like cousins. They were not on unspeakable terms. They were friends. You know, yeah, there's a little bit of war going on, you know, but we're cousins. And this close inbreeding led to the accumulation of a particular mutation called hemophilia, caused hemophilia A, deficiency of factor 8. A lot of bruising, a lot of blood loss. And if you have a lot of bruising, that means a lot of inflammation. And Russian prince several times almost died because of bruising and bleeding. Interesting fact about hemophilia A is that it is a sex-linked recessive condition. It is found on the X chromosome, Y chromosome, well, X chromosome should I say. So women are carriers, but only males get sick. Unless you purposefully breed sick male and carrier female, but that didn't happen. And it's interesting to see there's a scheme somewhere, you can find it online, hemophilia in the European royal families. You can see how genes for hemophilia got distributed over the main houses. So for instance, Germany and Russia, they were affected. But for instance, Spain and Britain, they got spared. Basically, it was a classic bottleneck effect in evolution and genetics. When new population is started with just a few organisms, whatever genetic information those organisms have, it will be passed on to the future generations. So at some point, you know, breeds got lucky. They didn't have any hemophiliacs, and they started to breed with commoners, basically, you know, increasing the genetic pool, essentially. So they got lucky, Spaniards got lucky, Germans and Russians not so lucky. So there was a lot of hemophilia going on. Okay? Now, terminology-wise, what I want to mention too. Intrinsic pathway, extrinsic pathway, common pathway. What happens at the prothrombin activator formation stage, that's the start of common pathway. That makes sense? Questions? So we're going to talk about anticoagulants and some other blood-related things on Wednesday, and I will see you then.